Many of you may know that this is the International Year of Crystallography, in which we celebrate the many accomplishments of crystallography to science. And in particular, we're today celebrating the contributions to biology. And one of the, this is the calendar that was put together by the PDB, WWPDB, to commemorate this important year of crystallography. And I think it's very appropriate that the September calendar highlights reverse transcriptase. So Eddy Arnold is an example of a crystallographer, structural biologist, who has done all of us proud in highlighting the very best of crystallography and its contributions to science, biology, and medicine. So Eddy started out at Cornell as a chemistry major, right? With John Collardi, where he did, John was one of the premier small molecule crystallographers. And then Eddy did some very wonderful work at Cornell, and then went to Purdue, where he worked with Michael Rossman, and never wanting to do anything that wasn't a great challenge, he took on the structure of rhinovirus in the 1980s, when that was really innovative kind of work. He then came to Rutgers, to the CABM as an assistant professor, and again took on an enormously challenging project to understand the structure of reverse transcriptase, which he did. And through the years, he has, as they say, risen through the ranks. He has been honored in many different ways. He has one of the few people, I think, in the United States who has had 2 merit awards from the NIH in these times when getting any money is very difficult. I was very pleased to be invited to a wonderful party to celebrate his receiving of an award from the Hyacinth Foundation because of his very important work with respect to HIV/AIDS. So Eddy has done wonderful work in using the very best there is in structural biology to be able to actually do drug discovery. And today he's going to tell us about this work that has been the hallmark of his career. Thank you
so much, Helen. Thank you all so much for coming, and thank you for the really wonderful introduction, Helen. I'm really flattered. Today what I want to do is tell you about some of the odyssey of the studies in reverse transcriptase and some of the history of it, but really focus on the biology and the kinds of insights that we obtain. And as Helen said, we began on this project right at the beginning of our work. At the very beginning, though, as part of a little quiz for the International Year of Crystallography, I'd like to ask you, do you know what famous scientist in the 19th century discovered the phenomenon of stereochemistry using crystallography? This person might be unexpected to most of you. If you're under 25 years old, raise your hands if you know the answer. This is a person who started out in physical organic chemistry, using things like measurement of polarized light, see about circular polarization. How about people under 30? Can you raise your hand if you know the answer? Okay, 35. I'm looking for young people to know this answer because it's a real crossover between crystallography and medicine. Whoops, how about How about if you're under 40? So anyway, Louis Pasteur, who's of course best known for creating the germ theory of disease, actually discovered the phenomenon of stereochemistry by looking at tartaric acid crystals that had shapes that he could only superimpose by sort of relating them by a mirror image. And he thought about this a lot. He was really wonderful in his use of a microscope as a tool for observation. And in fact, he made a tremendous breakthrough at the age of something like 25. This was during his PhD thesis work in the mid-1800s. So I think this is the one of the most wonderful discoveries in the history of crystallography. And to me, it's special because, like Pasteur, I've really invested a lot of my effort in trying to understand how microbes work and how infectious diseases work, and in finding ways to both treat them and prevent them. So I think that's a neat connection. So I'm going to tell you that reverse transcriptase is an essential enzyme of HIV. It's the target of 13 drugs. It's actually one of medicine's most central drug targets. Reverse transcription is an acrobatic enzyme. It actually carries out 20,000 nucleotide incorporation steps in copying this single-stranded RNA genome. It does that virtually unassisted. Structures are known of RT in multiple functional states. RT conformational changes are critical for function and for drug opportunities. And so, that'll be actually part of the theme. Nucleoside analog drugs are mimics of normal nucleotide substrates, incoming DNTP substrates, but typically they can't be elongated. They poison the process even though they become incorporated. Reverse transcriptase structures have enabled the development of two non-nucleoside RT inhibitor drugs that have overcome resistance. This is work that we've done with the late, great Dr. Paul Janssen. And through this work, we developed an idea called the strategic flexibility concept, which has to do with conformational flexibility and positional variation of inhibitor that can help in overcoming resistance. And this we refer to as the strategic flexibility concept. And we've, in the past 5 or 7 years, been able to engineer very high-resolution reverse transcriptase crystals. This has enabled a whole series of studies, including a new drug discovery approach called fragment screening. And in fact, in a recent set of studies from Joe Bauman in our group, I'll talk briefly about a new crystallographic structure determination approach using brominated fragments, 4-bromopyrazole or iodopyrazole, to obtain anomalous phasing from diffraction patterns that can be used to solve crystal structures very, very rapidly. At the beginning, I wanted to tell you a little bit about HIV and its relationship with AIDS. As you all know, HIV causes AIDS. Untreated HIV infection usually leads to death, not from HIV itself, but from other disease agents that take over when the immune system is eviscerated. A key aspect of HIV, first of all, it's a human retrovirus. It mutates rapidly. That means that in the process of copying a 10 kilobase RNA genome, there's about 1 change in each replication cycle that occurs. And when you put together this information with the fact that there are more than 10 to 100 billion virus particles being created in an infected individual every day, with about 2 days per replication cycle, what this leads to is a swarm of viruses with an individual that's often referred to as a quasi-species nature of viral population. And this is very complicating for either therapeutic targeting or prophylactic blocking, such as through vaccine development, because basically HIV is a moving target. This leads to resistance in the case of drug development. It leads to low immune cross-reactivity in the case of vaccine development. This is a very important concept, not only for HIV, but many infectious diseases caused by agents. The frequency of mutation can vary dramatically, but it's these changes that lead to problems down the road, particularly if the therapeutic development does not take into account the variation. So most of the anti-AIDS drugs actually target the enzymes. And these include reverse transcriptase. You've already been introduced to a beautiful picture of reverse transcriptase by Helen. These are the different components of the virus, the proteins that are encoded by HIV. This is a lovely image by our colleague David Goodsell, who's at Scripps. And what it shows is a pictorial cartoon-like representation of the virus particle. It's got a lipid bilayer that contains the viral components, including enzymes, structural proteins, surface glycoproteins that stud the surface and direct it to the appropriate target cells. And in the middle of all of this, there's a capsid-like assembly involving primarily the capsid protein that contains the 2 copies of RNA that are used for transmitting the complete infection to a target cell. And knowledge of the structure of these components, particularly RT and protease, has led to very considerable development of drugs through structure-based design methods. I'm going to focus on reverse transcriptase today, but I should mention that protease structural work has led to the development of some 10 drugs or so, which are very important, especially the most recent one, darunavir. Most HIV is treated with combinations of 3 or more drugs. Usually there are at least 2 reverse transcriptase, sometimes 3 transcriptase inhibitors, and protease inhibitors are still used in something like 30% of all combinations applied. The key thing about the introduction of the protease inhibitors together with RT inhibitors in the 1990s was that the death rate started falling off dramatically because now the 3 drug combinations were sufficiently potent to block ongoing viral replication. So here's a different, here's also a cartoon-like representation of what happens when an HIV particle comes in contact with one of the T4 cells that have the CD4 receptor as some of the key cells that control the immune system. Basically, HIV targets and hijacks cells in the immune system. This is what leads to the immune system dysfunction and disastrous consequences when our bodies can't fight, common invaders. So HIV comes in, attaches to particular host cells. I didn't know that would go like that. In the process of attachment, this is followed by fusion, where the viral components are delivered into the cellular interior. This includes the capsid-like assembly. It includes enzymes like reverse transcriptase. And it's at this stage when reverse transcriptase gets to work, and in a complicated process involving jumps and all sorts of different things, including priming of the process by a cellular tRNA, reverse transcriptase ends up making a complete double-stranded DNA copy of the 10 kilobase RNA genome. It also uses another enzymatic activity called ribonuclease H in this process of copying, in which the enzyme degrades the original RNA. And again, it's this double-stranded DNA copy which gets then carried into the nucleus and integrated into host cell chromosomes. At this stage, this is a permanent infection, and that cell may then become a factory for the production of new RNA, for the production of proteins within that cell. Some of the RNA gets packaged into newly forming particles, and the protease enzyme that's translated from some of the RNA is used to process what are initially immature polyproteins that are translated from the viral messages to eventual mature virus particles that can go off after budding, maturation, and infect new cells. The integrase enzyme actually is an enzyme that allows the double-stranded DNA genome to be inserted in the host cell chromosomes. Reverse transcriptase, integrase, and protease are three of the most important targets of the current 26 drugs that we have for treating HIV infection. Again, HIV programs the cell to make many copies of itself, and it's essential to understand that drugs target the machinery of the virus, including RT, reverse transcriptase, and protease. Shown down here is a picture of reverse transcriptase that we solved in the early 1990s, as Helen reflects back to when I was an assistant professor. I was crazy enough to work with some people to try to solve the structure of RT, and we were very fortunate to solve the structure of RT with double-stranded DNA template primer. In fact, this was the first structure of any polymerase with DNA in a relevant position to understand catalysis. And so the information on polymerase structure was very limited at the time. Certainly amongst our major motivation was try to understand how polymerases worked. So this tiny little spot in a cell actually gets blown up into something. This picture is about 10 million times magnified from the actual size of molecules. Molecules are incredibly tiny. They move very fast. And again, among our major motivation was that if we knew this kind of structure, we could facilitate drug design against HIV. I should say that in the mid-1980s, the specter of HIV was unbelievably frightful to everyone. People were dropping right and left. Many new people were being infected regularly, and there was nothing available to stop the advance. In 1987, the first drug, AZT, was approved for blocking HIV reverse transcriptase, but that was not enough. People were sick again within 6 to 12 months, but that was a tremendous advance because it at least showed that there was some way to go about it. It required the development of very potent 3-drug combinations to really pull the virus under control. And since AZT, there's been a lot of progress. This is a slide from a few years ago showing some of the many kinds of medicines that are available, including the nucleoside analogs that target reverse transcriptase. There's some 8 of them, including combinations. Tenofovir is the most used of these. Non-nucleoside inhibitors, including 2 drugs that we've helped to develop through work with Dr. Paul Janssen, are among the most broadly used, especially Sustiva and Intolince. I'll tell you more about these. There are some entry inhibitors that block entry processes. There are by now 3 different integrase inhibitors that are very valuable. And then there's a whole series of protease inhibitors, including darunavir or Prozista, that's got exceptional potency. At this stage, I'd like to show you more about the molecular anatomy of reverse transcriptase, and especially how looking at different kinds of structures of reverse transcriptase, its different functional states, gives you information about how this molecular machine carries out its business, and also how we can use that information for drug design. So reverse transcriptase is initially translated as part of a long polyprotein that includes the protease, reverse transcriptase, and integrase. Actually, even more than that, it starts with whole structural proteins. But by the action of HIV protease, the eventual polypeptides that are in the mature versions are released. And in the case of reverse transcriptase, we have a P66 subunit. 3-dimensional structure is shown here. And it was known early on from mutagenesis studies from Larder and Hughes that there were critical aspartate residues that were seen to be evolutionary conserved in all RNA-dependent polymerases that were required for polymerase function, and also key residues in the RNase H or ribonuclease H function. The P51 subunit is a proteolytic product from P66. So these two subunits actually have the same amino acid sequence. But oddly enough, in the P66, P51 heterodimer, these are arranged in such a way that they're always in chemically different environments. So, for example, the polymerase active site residues, although present in P51, actually don't contribute to catalysis because they're buried. So the initial structures, first from the Stites lab with a 3.5 angstrom resolution structure with the drug nevirapine, and then from our lab with the structure of RT with DNA, showed a lot about what the function was going on. One of the salient features in the P66 subunit was that the fingers, palm, and thumb region resembled a human right hand, something that had also been seen from the Stites work on the structure of E. coli DNA polymerase 1 clinal fragment. So one of the unusual things here is that the RT-P66 P51 heterodimer is actually the most asymmetric unit, or the most asymmetric dimer that I'm aware of in biology, even though the two subunits have the same sequence. This is a different depiction of the structure, showing the P66 subunit in fingers, palm, thumb, color coding, connecting the polymerase domain with the RNase H. And what this shows is the relative location of the nucleic acid as it's being grasped by the enzyme, and also the polymerase active site shown in gold, and the non-nucleoside inhibitor binding site shown in cyan. So these are the two sites at which all of the known RT drugs block infection, block polymerization. The NRTI binding site is a hydrophobic pocket that's about 10 angstroms away from the polymerase active site. As we use crystallography to understand many different things about reverse transcriptase, the technology is really driven by the kinds of protein constructs we make, the types of crystals that we're able to obtain. And I'm happy to be able to share this with you to show, first of all, how lovely some of these crystals look, how beautifully many of them diffract, and how very diverse they are in terms of the kinds of questions we can ask. For example, we've got structures of RT with no bound ligand, structures with bound non-nucleoside inhibitors, numerous crystal forms with various nucleic acid-bound forms, drug-bound forms. And this is fantastic because it allows us to look in atom-by-atom detail at many of the relevant states, and we continue to search for more. I should mention in particular that some of the very high-resolution forms, 1.8 angstrom resolution, sometimes 1.5 angstrom resolution, these were really pioneered by Joe Bauman through his crystal engineering. These allow us to do very unusual things, including collecting thousands of data sets to look at all sorts of systematic studies. The initial structures from 3 angstrom resolution data sets, et cetera, took months and months and months to obtain reliable structures. But this is incredibly important in terms of technology, and this is allowing us to look at many different questions. What's shown here is the relationship between the enzymatic reaction cycle for reverse transcriptase and the various types of structural forms. And we have good crystals that describe all these different forms. And so, for example, the unligand enzyme starts out with its thumb down, a movement of about 35 angstrom of the thumb leads to its ability to bind both nucleic acid and also non-nucleoside RT inhibitors. And it's at this stage that the enzyme is poised to carry out polymerase catalysis, because this is the stage at which an incoming DNTP substrate can bind to form a catalytic complex. The first catalytic complex was described by Steve Harrison from Harvard in 1998. That led to all sorts of further understanding. We've obtained many relevant structures of that type in the meantime. And this is the stage at which chemistry can occur, again, with some more functionally important conformational changes where the fingers now close down on the heart of the polymerase during catalysis. And then as catalysis occurs, this occurs with the evolution of pyrophosphate, and then the enzyme following the chemistry can then translocate and reload and carry out the process many times. We've also been able to capture the enzyme several times in the product form prior to translocation, thanks to specific disulfide cross-linking chemistry that we've done largely in collaboration with Roger Jones in our chemistry group. Very nice technologies to allow us to get key pictures involved in not only drug design, but also understanding the enzymology. So these are many different structures representing the NNRTI-bound form, the different kinds of interconversions that occur with, binding of NNRTI, binding of DNA. We're representing this as looking at the fingers, palm, and thumb subsection. Just in 2012, we obtained a very elusive structure where we had been looking to see how the non-nucleoside inhibitors, such as nevirapine, bind in the presence of nucleic acid. This was the first time after 20 years that we could actually clarify the mechanism of this type of drug's inhibition, and it took literally thousands of experiments to get to that point. So it was delightful when Callian Das and Sergio Martinez were able to get that to work in our group. So these are all very relevant and important functional states. These provide a lot of fuel for understanding via molecular dynamics, correlation with other biophysical techniques, NMR, many different features. And of course, I've talked a bit about function. I've talked about a bit about inhibition. We've also spent many years studying the specific mechanisms of resistance. Again, if a medicinal chemist aims to develop therapeutics in this area, unless they consider resistance, things are going to fail very rapidly. And all of these are input for drug design. These are the nucleoside analogs, or NRTIs. You've heard about AZT. 3TC is very important. FTC is very important. Tenofovir is a phosphonate inhibitor. And these are included, especially FTC and tenofovir, in the world's best 3-drug combinations that actually keep patients in great shape, basically the same life expectancy as normal, usually together with one other potent reverse transcriptase inhibitor, either an NNRTI, another RT inhibitor, or one of the other drugs that I've discussed. So in the non-nucleoside inhibitors that bind next to the active site, we have nevirapine, which was the first. Efavirenz is very important, very broadly used. And then we helped to develop 2 that are known as etravirine and rilpivirine. And I'm not spending that much time talking about mechanisms of drug resistance today, but this shows the locations of the positions that are important in causing resistance to the known drugs. For example, with one of the NNRTIs called TIBO, what we have is a picture of the molecule bound and then the locations of many residues that are involved in causing drug resistance. So for example, if patients or if cell culture are fed these drugs, there's so-called drug selection pressure that leads to advantages for mutants that can block binding. So for example, the tire 181 position, the lysine 103 position are common for leading to NNRTI resistance. The mechanisms typically involve straightforward organic chemistry mechanisms where basically steric hindrance that's introduced, loss of favorable contacts, loss of a hydrogen bond, and then some unusual things too, where the kinetics is changed through something like the opening rate of a pocket. So unusually though, in the case of the nucleoside analogs, we have a different situation. The nucleoside analogs bind at the active site, but there's really a more exotic, sort of array of positions there. One of the strangest things is that the mechanism of AZT resistance actually involves a ATP molecule that is subjugated from the cell to come in and actually remove AZT after it's incorporated. There are really diabolical mechanisms that occur in terms of resistance, showing how unusual the mechanisms can be by which a very desperate virus might be able to escape. So in terms of nucleoside analog drugs, of course, this is very intimately connected with trying to understand mechanisms of DNA polymerization. This shows the site of action at the polymerase active site, the 3 aspartates that live here, 2 metal ions that are involved in binding, and the relationship between the 3' hydroxyl and incoming DNTP. This has to base pair correctly with the template base, and incorporation typically only occurs efficiently if that's true. And so the 3' hydroxyl makes a nucleophilic attack on the alpha phosphate. You evolve pyrophosphate. This is the collection of nucleoside reverse transcriptase DNA chain terminators. These all require activation to their triphosphate form. Tenofovir actually starts out as a phosphonate, which is helpful because it's more easily activated to the triphosphate form. As I mentioned, 3TC, which has an unusual ring stereochemistry, PFV, which is acyclic, are really the 2 most broadly used agents, at least in the optimal circumstances. This is showing one of the considerations for what's going on here. This is showing a different schematic of this chemical process of incorporation, evolution of pyrophosphate, formation of the monophosphate diester bond at the 3' terminus of the primer strand. So in terms of how NRTI drugs are now, this is the non-nucleoside inhibitors. I'm going to switch over to that. How did these structures contribute to structure-guided design? Well, non-nucleoside inhibitors are very, very interesting molecules. They potently inhibit RT at nanomolar concentrations. They're chemically diverse. They're allosteric inhibitors, meaning that they work through locations other than the active site. And by now, there are 5 approved drugs of this type. Their structures are shown here. And one thing you can see immediately is that they're really quite chemically diverse. So nevirapine, dolaverdine, efavirenz have different chemical structures. And then the 3 most potent molecules that we developed, etravirine, rilpivirine, which are now drugs, and dapivirine, which is actually being used in a 10,000 patient trial for prophylaxis of HIV infection. These now have related chemical structures, whereas these don't. I'd like to introduce you to a wonderful human being that I had the great opportunity to work with. Our group worked with the great Dr. Paul Janssen for 13 years. After his passing in 2003, Belgium even put out a very important publication that honored him. He was honored along with the other greats in Belgian science, in this case for his development of drugs. And I was very, very honored when, in fact, our drug within reverse transcriptase was even modeled from our collaborative studies. Dr. Paul was very special. In 1986, I learned that Dr. Paul decided to target HIV/AIDS. In a parallel universe, I had met Dr. Stephen Hughes at the NIH, and we decided to target reverse transcriptase for crystallography. In 1987, Dr. Paul, together with Eric de Klerk in Belgium, started to search for anti-HIV compounds, and he found the potent and novel TIBO compounds, as specific inhibitors of reverse transcriptase. And at that time, our lab at Rutgers began crystallization, because I got hired for my position in June 1987. In fact, the first weekend after we got here, Gail, for standing Arnold, and I drove Frederick, Maryland, begin working on the project with Steve and his coworkers. We were that excited about it. It was just absolutely critical. In 1990, Dr. Paul published a key paper in Nature describing the TIBO compounds, which he called non-nucleoside RT inhibitors. And as an assistant professor, very unknown, just sort of beginning a lab, I sent a proposal to Dr. Paul, a 2-page letter describing our work and how I thought it might be useful to work together on some of his compounds. And, you know, there are 3 possible outcomes of this. Most likely is you just won't hear back. The second is, the answer is, no, you can't have it. The amazing thing was, I actually got a fax from Dr. Paul asking me when was the earliest that I could come over to Belgium and discuss the project with him. So I was so delighted, but I didn't actually know who I would meet because I didn't know that much about him. I thought, well, maybe I'd meet a person in a corner office in a suit, and then I'd spend a day talking with the scientists. Instead, the amazing thing was that Dr. Paul was just an absolutely immensely passionate scientist. Even though by that time he had already developed 77 drugs, and he had founded Janssen Pharmaceutical, one of the most famous drug companies, most successful drug companies ever, he just wanted to know absolutely everything about the project. He wanted to know how we were making the protein, what are the pH of your solutions, how do you crystallize something like this, but especially he wanted to know why would it be useful to know these structures, because I've developed 77 drugs and we haven't gone about it in this way. And so we had a very stimulating discussion. This was also Kun Andrews, who was one of my hosts, a virologist who had been spearheading the project. And even though Dr. Paul was very skeptical about this concept of structure-based drug design, he said, you know, let's work together on this. And that was fantastic because then we could really begin to try to use his compounds to help our work and our concepts to help his work. This man was truly an incredible person. He was an MD, he was a psychiatrist, he interviewed all the patients for his psychiatric trials. He developed 5 of the medicines that are still on the WHO list of essential medications. Just an amazing experience. Well, so as I said, fortunately, we really began working in earnest with him. And by about 1993, when we got our first RT DNA structure published, we had a good structure at the end of 1992. We were also working in parallel on Dr. Paul's drugs. So the TIBO compound, alpha-APA, which was another derivative, again, quite diverse chemistry, we were able to crystallize. This is in work from Kaoyan Das, Zhanping Ding. We were able to crystallize and obtain structures. What's shown here is the protein surface of the hydrophobic pocket that binds the non-nucleoside inhibitors. Some of the residues have been stripped away for clarity. But what you can see is that the pocket really makes quite a different shape depending on which compound it encloses. It kind of shrink wraps itself around the inhibitors. And this is in comparison to the Stites-nevirapine complex. So putting all this information together, we could see that, in fact, they had quite similar shapes binding in the pocket. They had somewhat of a butterfly-like shape. And that actually was very informative because then chemists began, for example, mixing and matching the different wings of these structures, leading to a great increase in understanding of the binding modes, the possible design considerations there. We also learned from these studies, shown in green here are the amino acid residue positions in this pocket region in the absence of the bound inhibitor, and then how they swivel and move dramatically away from the site to create the binding space. So there's no pocket in the APOS enzyme, and even then, we thought that conformational breathing or conformational selection had to be going on because these changes were simply too large to be accounted for by anything else. Even 20 years ago, we knew intuitively that that had to be what was going on to create the pocket, but it also emphasized that you really must know the structure with the bound inhibitor to have any chance of design. Otherwise, the design prospects are absurd. This summarizes a lot of the elegant chemistry that occurred following these stages. Those were initial structures. A really key breakthrough occurred when a series called the isothioureas were redesigned with the Pasteur concept to replace this double-bonded sulfur. And the resulting compound from here was completely unexpected. It actually crystallized in the reaction. And after the chemist tested it, the virologist tested it, all of a sudden this had by far the best potency of any of the compounds made to date. And the critical thing was, and it's called a data compound because it has a diaryl triazine structure. The critical thing about this is that it was incredibly simple to make. It could be made in one step from readily available starting materials. So all of a sudden they can make hundreds of compounds, even just starting from compounds from the literature. So this breakthrough in 1994 led to a very active design period. Callie and Doss, I, others in the group went over to Belgium many times to interact with Dr. Paul. Now it is Center for Molecular Design. By 1993, he was so excited about the structure-based drug design against reverse transcriptase that he created an institute called the Center for Molecular Design, where he spent 90% of his research effort working on this design project. This was very gratifying for me since he had started out very skeptically about the prospects. Some beautiful breakthroughs occurred over the period from 1994 to 1998 through a lot of computational design, continuing medicinal chemistry, but especially inspired by the computational design. The designers suggested a pyrimidine nucleus. They suggested removal of the exocyclic amino. Chemists were a bit reluctant because it required complete retooling of the synthesis. But once they made this compound, it just blew everything else away that had ever been made. This subsequently led, these are the DAPY compounds with diaryl pyrimidine nucleus. You can see that they're quite different substitutions, but this led to etravirine and rilpivirine as the actual approved drugs within a year or two. This shows in brief the drug design cycle. We were involved in obtaining the best compounds, determining crystal structures. That information was used to drive molecular modeling, to inspire further synthesis. They also had extremely good antiviral screening, initially at Janssen, but then at Thibotech, a company that was spun off Rudy Powell's Marie-Pierre de Pétune. And this was very active, using both the wild-type structures and the mutant structures. I believe they were the first group to do very thorough searches computationally of all the different mutant structures. And then the best compounds eventually showed to be successful in clinical trials. This shows another stage of the structure determination, now using the better compounds, including etravirine, one of the drugs eventually. And one of the things you can see here is now that we have quite similar structures, oddly enough, the pocket rearranges, the compounds rearrange, but there's quite a bit of flexibility for these compounds, even though they're quite closely related chemically. And what this led was to a hypothesis that Callie and Doss crystallized in a publication that we refer to as the strategic flexibility hypothesis. We had seen that conformational changes in flexibility in the inhibitors could help, but what we proposed was that some of the mechanisms, such as steric hindrance for blocking the activity of some of the earlier NRTIs, in fact, might be fixed if you have a flexible inhibitor, so that part of the molecule can kind of move out of the way of things that lead to a complete breakdown of activity. Also, if the compound is relatively compact, it can move around to get out of the way. And we refer to this as wiggling and jiggling as very possible favorable features of the compounds. You don't want to have too much flexibility because there will be too great an entropy cost, just the right amount. And these compounds really just were very superior in terms of their activities, even against patient-resistant sera. And especially the discovery of rilpivirine was revolutionary because now it actually had another flexible group, the cyanovinyl functionality. And if we look at the scorecard of how these compounds work against wild-type and drug-resistant viruses, take nevirapine, for example. It's still used in the developing world. It has catastrophic resistance even with a few mutations. You get 10 to 1,000-fold resistance just from a single mutation in the virus. It doesn't take much to imagine, given the combinatorics of mutation, that those are even present all the time. So now you get to efavirenz. It's really got very good properties, especially in combinations. It's got difficulties against some of the common resistant mutant, anywhere from 30 to 50-fold resistance. You get the rilpivirine. You have subnanomolar activity against all the common resistant variants. So that was really a tremendous breakthrough, and Dr. Paul had this as his favorite molecule. It's pictured on the stamp in many other places. We also learned some very interesting things that I don't have time to tell you about, where the compounds probably are delivered as hydrophobic aggregates into the cells. But, the bottom line is that these are not only potent in cell cultures in animal, but they actually led to 3 approved anti-AIDS medications. Etravirine was approved as Influenza in 2008 for treating patients that are already resistant to prior NRTI drugs. And then beautifully in 2011, rilpivirine was approved as Edurant in 2011. This is actually for drug-naive patients. The amazing thing here is that the dosage is 25 milligrams a day versus, say, 600 milligrams for efavirenz. So tiny pills, even these once-a-day triple combinations. Here's a 525 milligram pill that if somebody can take adherently, they can actually be healthy for the rest of their life unless there's some other complication that comes up. Rarely some resistance does get selected, usually having to do with the N-tricidabine part of it, the 3-TC part of it. One of the most gratifying things to me is that Johnson & Johnson, who markets these drugs, has granted royalty-free licenses to make and sell the generic drug at cost in the developing world. This means that millions of people can benefit from these drugs, and that's certainly extremely gratifying. We've since that time, since the drugs were developed, we've undertaken a whole series of studies to try to understand how they work. Is this wiggling and jiggling correct? And in really brilliant work from Joe Bauman and Kalyan Das, we were able to show that because of the wiggling and jiggling, rilpivirine maintains its activity against these common resistant variants. So the drug moves, for example, by 1.5 angstroms in the pocket by the change from an isoleucine or a leucine to an isoleucine, evading steric hindrance. Here we see wiggling where the cyanovinyl is moved 45 degrees to basically maximize interactions in a changing chemical environment caused by the mutations. I'm briefly going to tell you about, I still want to take a few minutes at the end to tell you about this new phasing method, but there's a flexible crystal form of rtDNA that Kalyan Das and Sergio Martinez have put to great use. And as I mentioned, for the first time a couple years ago, we were able to publish the structure of RT plus DNA plus the drug nevirapine. This is the first time that had been accomplished. We had actually predicted a while back that the binding of the drug would actually cause a piece of the structure called the primer grip to be displaced from the active site, and that it might actually move the nucleic acid out of the active site. It turns out that that is exactly what happened. So, for example, show you morphing. So small here, I'm sorry. Oh, gosh. Oh, not working on this computer. I'm sorry about that. So, in fact, what happens is the structure morphs between the normal position of having the primer terminus at the active site, but when the drug comes in, it actually gets displaced 4.5 angstroms away from the polymerase active site. And this is a beautiful demonstration that the distortion of the nucleic acid complex actually takes part of the chemical equation away from the catalytic site. So another important insight. What I'm going to do at this stage is briefly change over to tell you about a really terrific new development in our work from largely created by Joe Bauman. So one of the things that we were able to do with the reverse transcriptase, I mentioned that Joe did some protein engineering to improve the quality of the crystals. The story is that Joe was quite far into his PhD work, and several of the very ambitious things that he had tried to do, like clone the hepatitis B virus polymerase, the human capping enzyme, had led to all sorts of wonderful constructs, but no useful crystals, no structures, and I asked him at that time to try to engineer reverse transcriptase so we could get a good structure with rilpivirine. And I thought 3-angstrom resolution structure would be good, because for some odd reasons, they only diffracted to about 8. And in fact, he ended up getting 1.8 angstrom resolution crystals with rilpivirine, called me from the synchrotron and said, Eddy, how about 1.8? I had no idea what he was talking about, because that couldn't possibly be what we were most interested in. But it turned out that he was able to get a structure. That's how we knew the work with the structural flexibility was correct. He also subsequently, with Disha Patel, undertook a massive fragment screening effort where you take small drug-like fragments, soak them into the crystals, and find out in an unbiased way where they bind. What are potential drug binding sites? Where do ligands like to bind on proteins? So in that work, which was enabled by the high-resolution crystals, here's an example of a fragment that actually bound at 11 locations in reverse transcriptase. We were able to bind things to the known drug binding sites and actually all over reverse transcriptase. And it turns out that, we were thinking, gosh, this would be a great method for, determining all druggable sites on a given protein just by using this as one of the soaks. 4-bromopyrazole has very interesting properties. It's small, it's aromatic, it's got a halogen, meaning it can make halogen bonds, it's got a hydrogen bond donor, it's got a hydrogen bond acceptor. But then Joe went to think, maybe why not use this for crystallographic phasing? And most of you know that anomalous dispersion is one of the most powerful ways of solving crystal structures. Using especially synchrotron radiation, you can get signals from especially heavier atoms. You're most familiar probably with selenomethionine. People ask you to make selenomethionine constructs if you're a biochemist and you're working with a structural biologist. The reason is that they can really light up if you use certain choices of conditions for crystallography. Sulfur phasing is becoming the rage, and you will see that coming up. I won't tell you much about the physics of diffraction here, except to tell you that this kind of slide and what it tells you about elastic scattering and the relationship between different reflections in a crystal structure that are normally equivalent break down when the energy of the X-ray is close to some of the inner shell electrons. This leads to something called anomalous dispersion. Especially at a synchrotron, you can find energies to do this for almost any atom. At a home source, you can do this very, very strongly even with iodine without any fooling around with the radiation source. But it's this anomalous scattering where, in fact, the resultant scattering from something that causes different changes for what are usually the same. These are called Friedel pairs, and when you have anomalous scattering, they're called bifoot pairs. The key thing is that you can cause certain atoms to light up much the same way that you use mercury atoms or platinum atoms to light up in just about any kind of so-called heavy atom derivative. So a lot of crystallographic gobbledygook leads to an ability to combine current methods. The most important result is that when then Joe applied this idea, there were 11 binding sites in reverse transcriptase. He then searched a number of molecules that might do this. These are incredibly soluble in DMSO, 6 molar, 4 molar solutions. These are about half these molecules, half DMSO. Joe soaked crystals, submerged them in liquid nitrogen, collected X-ray diffraction, for example, at a synchrotron, at a good place for bromine. And he also did smart things in terms of the data collection to minimize systematic error. And of course, if you want to do this sort of thing, you want to understand some of those things. But the most incredible result was that when he took a crystal of one of our favorite proteins that we work on, called the cap-snatching influenza endonuclease, he soaked them, in this case, for 500 millimolar using bromine, 10 minutes, collected a data set at the synchrotron, with some smart methods of data collection, but, something routine. And 30 minutes later, from a program called AutoSolve in Phoenix, he had a complete structure that was refined, solved by magic, and he didn't have to make any kind of derivative. These were just the crystals that he liked to work on. And 185 out of the 187 normal residues were actually refined and completely visible from this. He ended up already with a publishable structure immediately. An interesting thing is that there were actually 25 sites located in the structure, a very reasonable figure of merit. This is all from the anomalous signal from the bromines in the solvent. And in a more crystallographic environment, we can go over this in great detail. Data was collected very carefully. He also applied this. So here's the 4-bromopyrazole. These are the locations of the anomalous peaks. In this case, there were at least 3 of the peaks corresponding to the bromopyrazole. Turns out these smaller peaks come from the anomalous signal from sulfurs and also from 2 metal ions that are present in the active site. Here's the result using rotating anode detector downstairs at home, no synchrotron, with 4-iodopyrazole. And just a beautiful result. Very strong anomalous signal. It turns out that there are about 7 anomalous iodine electrons for signal using copper. This is the setup that we have at CABM. So here we have the scattering now from 6 iodopyrazole compounds. And again, this gave just an absolutely perfect solution right off the bat. So another test case, which is one of everyone's test cases, kind of like lysozyme, because it grows perfect crystals easily, diffracts incredibly well. This was also measured using 4-iodopyrazole, using the rotating anode Rigaku Micromax 007, copper K-alpha radiation, RX is 4+ detector, now collecting data to 1.45 angstrom resolution, gives just an absolutely beautiful solution, complete structure, 278 out of 279 residues in only 30 minutes after data collection. Now, people have actually developed related methods where they've used, say, potassium iodide soaking to solve structures like this. And also, Sheldrick used an organic compound that's magic triangle. It's tribrominated, triiodinated. Unfortunately, that's not commercially available. So bromo and iodopirazole, very safe when compared to many heavy atom derivatives. Usually you have to work with mercury, lead, platinum, these things that are quite toxic and even hard to get these days. Synchrotrons don't like you to bring them. They're very inexpensive and commercially available, about $50 a gram. And it's fast and minimal labor in methodology, especially compared to SeleNoMep. So we're really excited about this method. We want to apply it to unknown problems, trying to understand it better, solve each of these multiple times so that we make sure that the uniformity is good. But very innovative work from Joe. At this stage, what I'd like to do is thank you all very much. I need to acknowledge, however, my coworkers in the lab. This is work that's really been spearheaded at many stages by Kalyan Das, by Joe Bauman, by Sergio Martinez, by Steve Tusky, by Disha Patel. And I've tried to mention other people as they've fit into the picture. I've mentioned Dr. Paul Jansen and his fabulous group that really did the drug development based on our collaboration. Our collaborator, Stephen Hughes, has contributed to so many different aspects of the studies. And also in recent work with Karine Musier-Forsyth, Stefan Serafianos, Mike Parniak, we're working on the RT initiation complex. We have some very exciting new results on that. Work spearheaded by Steve Tusky and Gary Joe Harrison. So at this stage, I thank you all so much for your interest and for your attention, and I'm available for questions.
So I'm curious about the flexibility leading to evasion of resistance. How general is this phenomenon? Has it been observed in other systems? And does it have to do with the flexibility on the protein side as well? And do you have to sort of match the flexibility in some way? And then finally, I guess maybe this is a bit too difficult in a way, but do you have any insights about how one might go about designing these kinds of molecules to incorporate this flexibility?
Actually, the answer is yes for all of those questions. The simple answer. Yeah, we do have numerous instances where flexibility clearly is helping compounds evade, resistance or at least ameliorate. And for example, in 1998, we published a paper where we saw some flexibility in an inhibitor complex and knew at that time that that could be very favorable, but really this multiple set of structures that Kalyan Das had in 2004 just really was very, very compelling. That's a very highly cited paper. It seems to have inspired quite a few groups to work in that direction. Often when I'm hearing people give talks on resistance, they talk about this as one of the components of what they're looking at. Celia Schiffer at the University of Massachusetts has created sort of a complementary approach that she calls the substrate envelope approach. And what she does is she creates a superposition of all drugs that bind in a particular site and then uses that to sort of design inhibitors that don't protrude through that substrate envelope. And we had published similar concepts, but she really gave that eloquent description. And even there, for example, Celia Schiffer will say, yeah, some wiggling within the envelope seems to be a good thing. As far as the complementarity with the protein, that turns out to be a very interesting dimension of this. We saw, for example, in the high-resolution structures of our drug rilpivirine with RT drug-resistant variants, that in fact there was quite a bit of shift and accommodation of the proteins. So again, in that case, you see some wiggling, you see some jiggling, and I refer to that as a shrink wrap effect, where this flexible pocket kind of makes largely improved van der Waals kinds of interactions. So all of these things are in play. As far as developing an algorithm, I mean, for 10 years I've had a very clear idea of how to do that, but we just haven't implemented it. You know, one of the connected concepts of this is that drugs can have multiple, you know, stable conformations, and that's what we're talking about here. We're not talking about the drug getting into a strain conformation, because people have seen that that usually leads to, you know, reduced activity. You want the drug to be still comfortable after it's wiggled a bit to now accommodate to a drug, you know, to a mutation change. Well, and that's why you don't want too many, you know, flexible bonds. So some people say, well, you know, if you have increased flexibility, you blow it because you'll have too much of an entropy cost. But it's a Goldilocks phenomenon. These compounds have 4 or 5 rotatable bonds. They don't have 10. You know, I'm not advocating putting in a methylene linker to achieve flexibility.
Nucleotide analogs that they use protective groups over those phosphates as a way to get into cells. I just wondered if people have gone back and revisited the nucleotide analog drugs for HIV to see if that is an effective way to improve their delivery.
Well, that's in fact where the idea comes from, pretty much, because, for example, tenofovir is usually formulated as diisopropyl or diisoproxyl fumarate, and what it has are phosphonate esters or carbamate esters to the phosphonate that actually protect it before it gets into the cell, facilitate the entry, and then they get removed. But yeah, these are very important. So if you have charged drug candidates that have wonderful potency, but they're not getting into cells, prodrug forms are really, really important.
Eddy, is RNase H drug target?
Sure, and I didn't have enough time to talk about RNase H today. Yeah, we've been pursuing RNase H inhibitors as potential drug candidates, especially in collaboration with Mike Parniak over the past decade or so. And in fact, we have some very potent RNase H inhibitors, but very rarely do they have antiviral activity. And almost invariably, when they do have antiviral activity, they turn out to be integrase inhibitors. And the reason for this is that the ones that are targeting the active site target a 2-metal ion mechanism that's very common. It's common for these polymerase active sites. It's common for our cap-snatching endonuclease active site, common for ribonuclease H active site. And so you have to contend with the different 2-metal ion active sites. In our collaboration with you, there's a 2-metal ion active site at the, the mitochondrial polymerase, RNA polymerase. But RNase H is still a viable and interesting target. One of the problems is that that region of the structure has a relatively flat face. It turns out that these pockets in structures are really wonderful for drug targeting because the pocket usually has features that you can target, and you can achieve the right balance of sort of hydrophobicity and hydrophilicity to end up with an actual drug molecule.
Okay, I would like to thank you very much, Eddy.
Thank you so much, Helen.
So the advantage of this